Welcome to the Dendrite Podcast with us, Saeed and Farnam, where we help students explore ongoing research and get involved in possible opportunities. This is a conversation with Dr. Steffanie Strathdee. She's an infectious epidemiologist, a Harold Simon Distinguished Professor at the University of California, San Diego School of Medicine, and a co-director at the Center of Innovative Phage Applications and Therapeutics. She is known for her work on HIV research and prevention programs and her advocacy and work on phage therapy.
Dr. Strathdee was one of the 50 most influential people in healthcare in 2018 by Time magazine, and in 2024 was listed as a top female scientist by Research.com. Steffanie and her husband, Dr. Thommos Patterson, are also best-selling authors. Their book, The Perfect Predator, A Scientist's Race to Save Her Husband from a Deadly Superbug, published in 2019, is a fascinating story of scientific innovation, love, and a will to life. We hope you enjoyed this episode, and now here's Steffanie Strathdee. Hello, Dr. Strathdee. How are you today?
I'm doing great. Nice to see you.
Thank you so much. Thank you for being a guest in our podcast. I'm just want to basically let you to explain a little bit of how this whole thing got started in the first place, and what are the some cool stories that you can share with us today about how this whole thing got started in the first place.
Sure. Okay. And my cat Benita says hello. So she always knows when I'm doing something on Zoom and has to insert herself. So hello, Benita. So I'm an infectious disease epidemiologist. I was trained in Canada. I'm both Canadian and American. I should say I'll be spending a lot more time in Canada after last week's election. So you'll hopefully be seeing more of me in person rather than just virtually. I have focused most of my research on HIV prevention, both in Canada and the States, and most recently in Mexico. However, my husband fell ill with a terrible multi-drug resistant bacterial infection while we were traveling in Egypt in 2015. And he was medevaced first to Germany, then back to San Diego, where we live right now. And he was dying, and my colleagues were the ones caring for him. And when they told me that they'd run out of options because it was pan-resistant, so resistant to every antibiotic that was available, I did my own research and found a 100-year-old forgotten cure, which are bacteriophage, which are viruses that have naturally evolved to attack bacteria. And I learned that they had been used to treat bacterial infections over 100 years ago. But they fell out of favor in part because antibiotics came on the scene. And so clinical trials to show that they're efficacious had not yet been done. So it was an experimental treatment. I was able to get approval from my colleagues to pursue this. And we sourced phages from around the world from researchers and even the US Navy, who provided phage that matched his bacterial isolate within 3 weeks. And although he was on the cusp of dying, he turned around and lifted his head off the pillow and kissed his daughter's hand when she was there on shift, and has made as near a complete recovery as possible. So that has ushered in this whole era of phage therapy in the West that had been forgotten. And now there's over 90 clinical trials on phage. And we've treated lots of other patients, and there's even a Phage Canada now. So it's a whole new field, and that's the short of it.
The side of the scientific side and personal side of this story just makes it fascinating to listen to, and it's so inspiring. So I just wanted to ask you about, because you mentioned in the book that in your trip to Egypt and even in the previous trips that you had, you yourself did have the handy antibiotics to use in terms of anything that can happen. So what can you say now about the overuse of antibiotics in general? So how should we be thinking about using them?
Yes, well, I mean, when we decided to write our book, because if I was blindsided by the global crisis of antimicrobial resistance, or AMR, as we refer to it, then the average person probably is too. And most people are not aware of phage therapy. But, I was unwittingly contributing to problems of misuse of antibiotics, because they should always be prescribed by a physician to a patient, or a veterinarian if it's an animal. But it's really our misuse of antibiotics in livestock and agriculture, in animal husbandry, like with cows and chickens and pigs. That's really been the driver of AMR worldwide.
Mhm.
That is great. It's just amazing to hear that story and how you shared it with the whole world. And I'm sure our audience can go ahead and listen to it or listen to the whole story in the TikTok that you put in the YouTube. But I'm just wondering, so would there be a chance for phage therapy to completely change the word and instead of antibiotics, like take over the antibiotics completely, or you don't see it?
Well, I mean, my own personal belief and that of my colleagues at our center, because we now have a Center for Innovative Phage Applications and Therapeutics, which is a nonprofit based at UC San Diego. And we don't believe that phage are going to entirely replace antibiotics, but they will be an adjunct to antibiotics if the clinical trials show that they're efficacious. And clearly, they can be sourced from the environment because their phages are found wherever bacteria are found. So in soil and water already in our bodies, there's lots of phage there. So it's relatively cheap to source them. And the issue is you have to purify them in order for them to be used safely. But nevertheless, phages could be used to replace antibiotics in agriculture and in farming and in animal husbandry. So there's a tremendous opportunity for us to reduce the global spread of AMR by advancing phage therapy.
So in the book again, before your own proposal of phage therapy, my question is, why didn't any other of the medical staff and the team think of alternatives, alternative kinds of therapy? And is it in the present time, do we have any other alternatives than phage therapy, or how is the lookout for alternative therapy?
Yeah, well, the paper that I found had a couple of lofty, ideas for treating the bacterial infection my husband had, which was Acinetobacter baumannii. One of those was like a UV light therapy, but that can only be used on the skin. Another would be to develop a vaccine, but that was obviously, if that happens, it's, decades away. And the physicians that were treating him in the hospital, I mean, they are overrun with patients that are, very sick. So even though this was a patient who was a friend of theirs in many cases, they don't have the time or the energy to invest on that. So it was really my doing that came up with the idea to treat him with phage. And I was just grateful that they were willing to embrace it.
Awesome. I'm just wondering, how did your background specifically help you to navigate through this whole problem and get you started or support you throughout the whole experience?
Well, I was trained at U of T, so I took a microbiology course. In fact, I had a specialist, BSc honors microbiology degree that I took from 1984 to 1988. And in my virology class, we actually learned about phages. I just never knew that they had been used to treat bacterial infections. And so when I stumbled across this paper that mentioned phage, I knew what they were. And when I started to read that they had been used successfully in some cases to treat bacterial infections in animals and humans, I thought, wow, wouldn't that be cool if we could do this? And then I realized, oh, wow, this is experimental. Why is it experimental if it was discovered 100 years ago? And then, of course, I realized that phage had fallen out of favor in the West. It was also embraced by the former Soviet Union around the time of World War II. So it was seen as Soviet science, which was done by the enemy. So that geopolitical bias really drove a lot of the difference between the way the East and the West approached phage. So yeah, it was just I guess the fact that I've been trained how to do a literature review and to think outside the box when your back is up against the wall. I owe a lot to my mentors at U of T.
Right. That's great.
So let's now shift gears to present. So how does IPATH operate, and how strong is the international community that's working on phage therapy?
Well, our center, IPATH, has two main purposes. One is to assist patients like my husband who or have no antibiotic options left, and to help them navigate through the regulatory process for an N of 1 treatment. We also are trying to advance clinical trials, because that's what's going to be needed to license phage therapy alongside antibiotics. In the US, that's the FDA, the Food and Drug Administration, that has to approve either individual cases or a trial. In Canada, it's a different regulatory framework. Other countries have other models as well. So IPATH is, is a center that's like several others. Phage Canada is based at U of T and McMaster, led by Dr. Greg German. It's going to be, rolling out other trials as well.
Right. Could you explain a little bit more of what is it actually happening in those institute? Are they just hunting phages and going through the data, or
Well,
more?
it depends. If they're a phage therapy center, then they're helping patients get the treatment. So infectious disease physicians are involved, and they're, having to source phages, usually from other labs, or maybe, a company, and finding that phage and ensuring that it's a match, and then it has to go through a process of purification to eliminate endotoxin if it's present, and then to figure out the dosing schedule, the routes of administration, and then getting approval from either the FDA or Health Canada or whatever authority is overseeing it to get them the treatment. There are lots of labs that are doing translational research or basic science research to optimize phage for treatment. Some of these are working on natural phage. Some of them are working on genetically modified or even synthetic phage. So the field is moving very quickly. For those of you who use PubMed, the search engine that is, to find papers, a mesh term for phage therapy was just like came into being in 2017 after my husband's paper was published about his case report. So it's still a very new field.
Right. That's amazing, and we'll look forward to actually have that kind of facility to also have something in Ottawa as well. So we'll definitely look forward to that as well. So I know you already talked about this a lot in your book, but I'm just wondering if you can briefly also tell us more about why phage therapy and not the antibiotics, and what are some of the advantages that phages have and other treatment don't.
Sure. Okay, well, one of the advantages of phage is that they are very specific. So they only attack bacteria that have a receptor that they match to. So it's like a lock and key, like an enzyme, and so any bacteria that doesn't have the receptor that they match to is not affected. So in that way, it's like precision medicine at a nano scale. Whereas antibiotics tend to have a broad spectrum approach that kills the friendly bacteria as well as the pathogenic bacteria. And we now know that that can disrupt the microbiome and cause dysbiosis, and that has other negative health effects. So, and phage obviously can be sourced easily from the environment, and you don't need a lot of high-tech equipment to, do a phage hunt. It's the purification process. And if you need to develop phage under good manufacturing practices, or GMP as it's known, then that becomes expensive. But it's something that, citizen science and student scientists can engage in, which is why I like the CPhages program so well, and the program that exists at the University of Ottawa as part of that.
Could you also talk about what are some difficulties that you have experienced with testing patients with phage cocktails, and what are some possible failures that can happen throughout the way?
Yeah, well, there are failures that can occur, and but they are like they can be circumvented. But one of the biggest challenges is that sometimes we only find temperate phages instead of lytic phages, and temperate phages undergo what's called lysogeny. It's a different life cycle than the lytic cycle, which actually kills the bacteria, and those virions that are produced in lysis go on to attack other bacteria that they match to. But in lysogeny of a temperate phage integrates its genetic material, usually DNA, into the bacterial cell DNA and then hits the snooze button, is the way I describe it in our book. And so it doesn't kill the bacteria, which is bad for phage therapy because we want to kill pathogenic bacteria. It can also carry AMR genes or toxin genes, often does. So that could be a detriment to the patient later on. And it can even collude with the bacterial cell to make it resistant to attack from other phages. So for all those reasons, you don't want to use temperate phages. Sometimes that's all we can find. So then phage that are temperate need to be genetically manipulated to make them lytic phage. And then sometimes resistance to the phage occurs because CRISPRs are part of the bacterial immune system that have developed specifically to, target phage and to avoid phage. But phage have their anti-CRISPRs. So this coevolution of phage and bacteria has been going on for almost 4 billion years. And so when resistance occurs in the bacteria, you either, have to circumvent that somehow. So having a phage cocktail that has different phages that hit different receptors is one way of making it more difficult for the bacteria to overcome that issue. But also, if you have a phage library with different phage in it that attack different receptors, and they've already been characterized and sequenced, and you know which phage go best in a cocktail or which phage might be synergistic with an antibiotic, then you can go back to that phage library if resistance emerges, like it did in my husband's case, and you can source that new phage to and go on that way. So, and I think machine learning would play a very big role in that, and I believe it will in the future.
So is there some way that we can use a temperate phages? Because in my own phage hunt, the one that I have is a temperate phage, and for a couple of months I've been having difficulties getting a high titer. And I also remember that in one of the cases that you mentioned, because the bacteria was so rare and you were not able to find that many phages, besides one lytic phage, you use two other temperate phages. So is it just like the only possible use? Is it just the genome engineering, or what other informations can we drive out of these temperate phages?
Yeah, well, I mean, it is a bit of a false dichotomy to say that there's lytic phage and temperate phages because lytic phage can become temperate
Mhm.
and temperate phage can become lytic. And we don't understand a lot of that decision process. But there's different techniques that have been used to convert temperate phage to lytic phage. One that was in the case that you just referred to was a predecessor to CRISPR-Cas gene editing. It was called recombinering, where the repressor gene was clipped out and forced that temperate phage to become lytic. There also, obviously CRISPR-Cas gene editing can be used as well, and that's what a lot of people are doing. But there's also directed evolution, which is, and in layman's terms, you can consider it as how to train your bacteriophage. So, for example, there's thought to be only a certain number of bacterial mutations that can arise in response to attack from phage. And if you kind of study those mutations in the laboratory, say if you have like serial isolates from a patient over time, and you see that there's different mutations that are arising, then you can select phages that are going to attack those bacterial mutants. And in doing so, it's much harder for resistance to emerge. And that was done successfully in a case that involved a patient who was a victim of the terrorist attack in Belgium. And she had a shrapnel embedded in her leg. She'd been treated with antibiotic suppressive therapy for 3 years, but it didn't cure. And that phage was available to attack all the mutants that had arisen. And so she was not only treated, but she was cured, and now she is walking, and she was about to lose her leg. So I think that that approach and using, artificial intelligence to, select phages on that basis is going to be the way of the future.
That is great. That's definitely the next step with um, deep learning and all the new technology
Yes.
available. So again, I was wondering that do we know what do we know now about the standardized dosages and concentrations at the safe level of endotoxins, or whether when the bacteria is lysed, everything gets released in the blood or the system?
Yes. So endotoxin, for those that aren't familiar with it, is essentially a lipopolysaccharide layer, which is part of the gram-negative bacteria cell wall. And when lysis occurs, that cell wall becomes part of the bacterial debris that it is part of the phage preparation. And so when you're growing up phage in sufficient quantity to treat a patient, that bacterial cell debris, which includes this LPS or lipopolysaccharide, is essentially an endotoxin that can harm the patient. Now, it's thought that you should try to remove as much endotoxin as possible, but the rule of thumb that the FDA has used is 5 endotoxin units per kilogram per hour based on the weight of the patient. And that's what we used in my husband's protocol, which is now published. And we've used that benchmark for other patients, and we haven't seen any untoward side effects.
Right.
And what is the regulations of FDA? Because that was also like another concern. Is the timing accelerated, or is it now more favored? What
Well,
is the
the FDA has to approve phage therapy on a case-by-case basis if it's an emergency IND. So that's like emergency investigative new drug is that terminology. Or they can, if it's not seen as an urgent case, it can be given an IND, so an investigational new drug. That's the designation that a new experimental treatment gets. Now, obviously, phage is not a drug. It's, viruses, if you consider them alive, are, they multiply. But that's the only regulatory pathway that the FDA has. So at this point in time. So they have to approve, a phage in order for it to legally be administered by a physician in the US. And Canada has a different pathway with Health Canada.
Right. So it is kind of interesting that how complex and big this whole field is, but I'm just wondering, as of now, do we have any other alternative treatment other than phage and antibiotics for the diseases that are in practice?
Well, there are people that are studying phage lysins, which are the proteins on the capsid of the phage. So if you think of it as most phages have an icosahedron head, and they might have a neck and tail fibers. And the lysins that are part of the capsid of the head, those can be used as treatment in and of themselves. It's just that they're generally only used for gram-positive bacterial infections, and they can only be used topically, because if they are injected, say, into the bloodstream, that there's different enzymes that could degrade them.
Mhm. Okay. Okay, cool. So now let's just shift gears a little bit and talk about the whole mentality and the way that you dealt with this problem in the beginning and throughout the whole experience. Kind of walk us through what were you thinking, who were you relying on, and stuff like that.
Well, I had a lot of help. I often get too much credit as being, this person who saved my husband's life. I mean, it started with me, but the most important person alongside me was Dr. Chip Schooley, who was the head of infectious diseases at the time at UC San Diego. And he agreed to be the chair of the protocol, which even if I was an MD, which I'm not, I couldn't be the chair of the protocol because I would have been in conflict of interest, right? It was my husband who was the patient. So Chip really played a key role in determining what dose, what route of administration, how often, how long to treat him. And all of this was brand new to him, even though he's, a very established and well-known infectious disease physician scientist, and he's a virologist. He'd never administered phage before, so he had a lot of homework to do. And I also, had to reach out to other phage researchers around the world that were willing to send phage so that we could see if they were matches. Texas A&M served as a command center for this. And then the US Navy got involved because they had a phage therapy program that was very nascent. They'd only used phages on, rats and mice and a few rabbits. But my husband became the large animal model, so to speak.
So one
Interesting.
aspect about your book that I personally really appreciate is the way that you talk about or infer to Dr. Patterson. It would be easy to refer to him as just someone who's now in coma and waiting to be treated, or worst case scenario, not to wake up again. But with the interlaws and the way that you refer to him, you really talk about the real importance of him willing to live, to continue living. I was wondering if you
Right.
could talk about his experience through the illness and the PTSD after that, and how is the
Yeah. So in our book, The Perfect Predator, we decided that, you know, as we got to understand what each other went through, because he was in the hospital for 9 months, he was in the ICU for 4 of those 9 months. He was in a coma for two of approximately of those 4 months in the ICU. And so we went through the same ordeal, but we had different experiences. His version of what happened was through a lens of coma and morphine and toxins and the cacophony of being in the hospital and being near death. So his interludes are in the book that really are from his own experience. So some of these are hallucinations. And I used to think of it as this my reality was the right way, and his reality was like this crazy thing that wasn't true. But now I've come to appreciate that that was just it was an alternate reality, right? And I respect it for what it is. When he came out of the hospital, he was haunted by these memories, and they were very intrusive. So if somebody asked him about, what he experienced when he was in a coma, he couldn't turn it off, and he would have nightmares. And so we sought counseling. Both of us needed counseling, and we had counseling together. And the counselor who treated him said, "You cannot purge these memories entirely from your being. They are part of who you are." So he's come to see them as just as real memories as, us sitting here having this conversation and thinking about it like tomorrow. Um, but he did have a treatment called EMDR, which is used for patients that have undergone trauma to try to kind of reprogram the brain so that those traumatic experiences are no longer, so threatening. And it's interesting that approach is similar to psilocybin treatment, like that is being used to deal with PTSD. So the PTSD now, we still get triggered occasionally. Like, he's had some other health problems related to the side effects of his treatment, not the phage, but the antibiotics. And also that, having just his body inside having been ravaged by the bacterial infection. So when he's been in the hospital, subsequent, like, he was in the hospital because he had COVID, and, he has scarred lungs. So he was a very high risk patient. They put him in a hospital gown, the same kind of hospital gown he had been with him for 9 months, and we both looked at it. And I said, "It's okay. We're going to get through this. You're not going to be in the hospital for another 9 months." But you do get triggered, and you have to be gentle with one another, because what he went through, I mean, I think I understand it, but he says there's no way that anybody could ever understand. Even the hallucinations, the way they're written in the book, he says they come close, but there's no way to really capture it entirely.
just the idea of being on the verge of life and death, and
Yeah,
the,
exactly.
the travel through the desert when the leaf
Yes,
is now dried, and it's just
exactly.
terrifying. And being in
Yes,
that state of mind constantly for a month, it's really a great deal to come over that. And
definitely.
I was wondering if you could speak about science communication and being the right person in the right time, because in the book it is so clear that well, you and all the other team, it requires a great deal of quick response and communication to the emergency situation that you are in, and being able to critically think about it and come up with a solution that is often out of the box. So how do you suggest developing this skill, or how can we even think about it? How can we define it?
Yeah, I think science communication is so important. And we wrote this book and published it before the COVID pandemic began. But I started to realize how important it was for scientists to be real people. So we wrote the book as Steph and Tom, and our PhDs are not included on the cover intentionally because we didn't want to create more of an us and them. We wanted to show people that we were real people and that we make mistakes and that we have, traumas like anybody else. And I wanted it to be readable for a lay audience. Like my mother read it and said, "Oh, now I think I finally understand what you do." It's like, wow, I've had, other students in, medicine and virology and public health and microbiology give it to their parents and that their parents go, "Oh, I get it now." So, and so there is a skill in trying to take a complex idea and explain it to somebody who might not have any background or might not even have finished high school. And I think that one of those skills is the power of metaphor.
Mhm.
So, for example, describing the Acinetobacter baumannii bacteria and how it evolved to try to avoid the phage. I talked about it as it has a slimy capsule on its as an outer coat, and that you can think of it as somebody wearing a sweater, and if someone takes that sweater off, they have a different clothes on underneath, right? Well, when the Acinetobacter baumannii dropped its capsule, it was like essentially taking its sweater off, and that's where the receptor was for the phage. So it became resistant to the phage, but in doing so, it resensitized itself to one of the antibiotics that it had been resistant to just a few weeks earlier. So that's a super hard kind of concept, but when you put it in the context of metaphor, like a sweater, people get it.
Well, the human aspect of your book really spoke to me, because as I was speaking to my friends, I was like, I'm about to interview Steph, because that's how you
Yeah,
refer to yourself in the book. But
yeah.
then I was like, put your manners on, Dr. Strathdee. So, uh, I was wondering, so before your trip to Egypt, you had a completely different line of research going on.
Right.
Did that entirely change your career path, or are you still in touch with your previous research interests?
Right. So my research prior to this whole phage therapy story was really HIV prevention, but it has morphed into HIV and related comorbidities such as hepatitis C, sexually transmitted infections, overdose, COVID, Mpox. I'm still active in those fields, and am that's where I'm trained. So I'm not a virologist and I'm not an MD. If I was earlier in my career, I probably would have retrained to be able to write a phage therapy grant or a grant involving phage, but that's not where my expertise is. So my role in the phage world now is one of a public face for our story. My husband and I both talk publicly on a regular basis, and um, we also are involved in fundraising, and myself, I'm involved in the administration of IPATH. I also am involved in, teaching and talking to students. But I often get questions that I can't answer because it isn't my field. So I guess I'm active in both of these fields, but in different ways right now.
Awesome. That is just great how you and your husband contributing to the whole world and communicating that science. But I'm just wondering, us as undergrads, specifically at U Ottawa, I know TMM students already have phage hunting program and all that, but if you want to tell an undergrad student like me in biomed, how could we contribute to this communication of this science and how we could help this whole science to grow more?
Well, I think that we all need to play a role in science communication. Myself, when I have PhD students that I'm, examining for their dissertation, I ask them to tell me what their research is about in, 15 seconds. And I sometimes say that I'm your grandmother, you have to explain it to me that way. Or I'm somebody from CBC or Fox News or something like that. I do the Fox News one if I'm especially, being really hard because if they don't like what you say, they'll just talk over you and, and it's a skill that we have to learn as scientists because there's a growing chasm between the general community and science right now. They don't trust science, that so many quacks are being pursued instead. And, that's one of the reasons I'm so upset with the most recent US election because, we're turning back time like 100 or 200 years to say, let's take the fluoride out of water or let's get rid of vaccine mandates. So we have to be able to describe to people like what a vaccine does, and that you see side effects that are so rare, but you don't see the millions of infections that didn't happen, and the deaths that didn't happen because those were prevented. So that's a difficult concept. So I think all of us need to practice it amongst ourselves. And things like blogs and podcasts and TikToks, that and Insta, like it makes it so much more accessible for people. And I'm really stunned that our book has been received so well, and it makes me want to do more writing in this area.
That's great. And I hope everybody follow through the instructions in the book and also this podcast as well. So that is amazing. I'm just want to now maybe talk about your hobbies outside of the research. What do you do when you're not solving problems of phage therapy and antibiotics?
Well, I love to garden. And I'm right now I'm in sunny Southern California, so I still, was picking tomatoes just yesterday, which is I was in Ottawa last week, so I know that it's a little cold there now. I like to hike. Tom and I like to bird watch. We still travel. We've been to 13 different countries since his recovery. And I do like to write. I had written a fiction book after The Perfect Predator. I finished it on March 4th of 2020, and I remember the day because the book was about a pandemic that from a virus that was found in space. And I had envisioned how a pandemic might unfold on Earth. And unfortunately, most of the things I predicted came true. So when I shared it with our agent and other editors, they all said, "Oh my God, like I'm on quarantine right now at home. There's no way that I want to read this book." And it's just sitting there. So I'm trying to think of if there's a way I could recast it and go back to it. But to be honest, I'm so busy with my two jobs right now that I haven't done it. But maybe one of these days.
Maybe I pre-ordered that book. I love the writing and the narrative of the first one. So
Well, oh,
yeah.
thank you.
I was wondering if you could share one of your like good memories that you made in the 13 trips that you had after the incidents. It's quite impressive. 13 is like 13
Yeah,
different
13
countries.
countries. That's true. Well, I guess one of the trips that we took was to go to Chile. And that they have this conference called Congreso Futuro, and it's kind of like a bunch of TEDx's that are done, and it's broadcast all over South America. So we spoke at that, and then as a thank you, the organizers of that conference, which were actually the Chilean government, they said you could either go to Antarctica on a free trip, or go to like the Highlands, like to see the observatory, which was like I think it was 16,000 feet, and you had to actually have oxygen like tanks and masks to go there. And I was just terrified for myself, but mostly because of Tom, and he insisted on doing it. So anyway, it
So
was very
expected of him.
thrilling to see this observatory with 22 different countries having all of these giant telescopes that were all kind of moving like as if they were sunflowers towards the stars. So yeah, I think that's probably one of the highlights.
Awesome. We are so grateful to hear your stories, and we would look forward to hear others people's stories as well in the near future. So, what would be the takeaway or the next step for our audience or researchers, or what is the next plan or what we are moving towards with this whole
Well,
phage therapy?
Well, I think that this field is a very exciting one. In Canada, it's even more nascent as a program, but there's a lot of work that needs to be done in basic science and clinical science, and also machine learning and bioinformatics. If people are interested, I wrote a review article with my colleagues that was published in Cell last year, that kind of maps out the past, present, and future. I do think that in the future we will be able to just get the genetic sequence of bacteria rather than having the bacterial isolate shipped, because that takes time and it's very cumbersome. And if you just had the sequence of the bacteria, you should be able to match phage to it, or even synthesize phage to it. So there's even a Canadian company called Cytophage that is working in this area, and one of the staff is actually a U of Ottawa faculty member, Marissa Azad. So there are experts around you that are able to help you get into this area if you want to learn more. And of course, hunting for phages is really fun. A CPhage's student at the University of Ottawa named a phage after me, which is one of the reasons that I decided to come and speak in person. So I'm just really thrilled that it's a program that can source phages that are now being used to treat people.
Awesome.
One thing that's really great about this line of research is that there's just a lot that can be done and to explore, and we can see that, like the potential to genetically manipulate and engineer phages that would directly target the bacteria that you don't even have to send the cultures. It's so exciting, and I guess that's for us to take up and continue in that line. So again, I'd like to thank you for being the podcast being the guest of our podcast today, and I'm sure us and the audience and anyone who is interested will be much more inspired, and hopefully will continue learning and saving lives. Hopefully, thank you.
Great. Thanks very much. Take care,
Thank you.
everybody.
Thank you for listening to this conversation with Steffanie Strathdee. In this episode, we discussed the book The Perfect Predator, Antimicrobial Resistance, and Phage Therapy now and in the future. If you'd like to learn more about phage therapy, we've added a link to IPAF's website in the description. Make sure to check it out.
This is a Dendrite podcast. To support it, share this episode with your friends and anyone else who might find it interesting. And be on the lookout for our monthly journal club meeting. And now, let us leave you with a quote from Steffanie Strathdee. When scientific advances, masterful medicine, and the will to live come together, the impossible becomes possible. Thank you, and hope to see you next time.